Hello everyone. We are starting today with a cycle which is called as glycogenesis. So glycogenesis, as the name indicates, it is the cycle which leads with the formation of glycogen. And glycogen is formed with the help of many molecule and number of molecules of glucose. So basically this is a polymer which is made up of n number of glucose unit. Its main site of synthesis are liver and muscles. Around 100 gram of glycogen is present in liver, and in muscle it is around 300 grams. Both are formed in cytoplasm. So before starting this cycle, we have to understand what is the structure of glycogen. So glycogen is a complex structure. First of all, it is a complex structure. It's a long chain of many of the glucose unit. That is a polymer. So it contains many of the glucose unit which are attached by means of a linkage which is called as 1,4 linkage. Now apart from this, it has many branches too. There are the many branches, and these branches also contain same glucose units. They also contains glucose unit, and they also linked by means of 1,4 linkage. Now the only difference is there is a branch point where the branching takes place. So this branch point contains a different linkage, and this linkage is called as 1,6 linkage. So basically in glycogen there is a complex structure. There is a complex structure, and this complex structure is made up of many type of containing many monomers, and these monomers are glucose. Now the glucose units, they join together by means of a linkage, and this linkage is 1,4 glycosidic linkage. Apart from this, there are also certain branches which comes from this. There are certain branches which comes from this structure, and these branches also contains glucose molecules. So again you see here, they are the glucose molecules which are in this glucose. And these linkages are again the same linkage, that is 1,4 linkage. The only difference, as I discussed earlier, it is this linkage, this part, where there is a branch point, and this linkage is glycosidic 1,6 linkage. So in this structure there is a complex structure containing many things. So this I think so it is clear to all of you. Now the next part, moving to next parts, is formation. So the how glycogen is formed. Its formation occurs in three steps. Number first is activation. In activation, what is there? There is formation of active glucose, and this active glucose is formed. The second portion is called as elongation. That means chain elongate, and the last portion is termination or formation of complex structure. So basically there are the three steps. We will discuss one by one. So first is activation, elongation, and formation of complex. So starting with this activation, activation is, there are the few steps in activation. These steps are, the first is glucose, which is converted to glucose 6 phosphate. Easy step. The next is glucose 6 phosphate is converted to glucose 1 phosphate. And last and final is its conversion to UDP glucose. So these are the total step in activation. So as we discussed, there are certain enzymes. Glucose to glucose phosphate requires hexokinase, and glucose 6 phosphate to 1 phosphate required phosphoramidase. And the last is UDP glucose phosphorylase. So these are the three enzymes. The important aspect about these enzymes is kinase are the enzymes which are required for addition or removal of phosphorus. So this is kinase. The next is mutase. Mutase is required for change in position. Change in position. As you see here, the glucose phosphorus group changes in glucose from 6 to first position. So there is a change also. Now coming to phosphorylase, this is a different, and UDP glucose is finally formed. This is called as active glucose. So first there is activation. Now I'm starting with second process, which is elongation. How elongation takes place. Elongation is easy step. It requires this process takes place as we have already discussed. The process takes place in the site which is called as liver. So in liver there is a glycogen primer which is already present. Glycogen primer is already present. For example, this is glycogen primer which is present in liver. Now this combines with UDP glucose, which is active form of glucose, and ultimately what happens, this primer is there and it combines with one of the molecule of glucose and UDP is released. Okay, so this glucose combines and the enzyme is glycogen synthase. And this linkage is, which is very important, the linkage is 1,4 glycosidic linkage. So as we discussed, we discussed first enzyme which is called as glycogen synthase, which is required to combine this UDP glucose to glycogen primer. And here UDP is released and there is addition of one of the glucose unit to this. Now the next step is this glycogen primer with one of the molecule of glucose, it elongates and there is formation of a long chain with the help of glycogen synthase. The enzyme is for the synthesis of glycogen. So it is called as glycogen synthase. So a long chain is formed where 12 to 14 units of glucose are attached. So this is long chain and this process is elongation. Now there is action of another enzyme which is called as transferase. So again from this 6 to 8 transfer units, 1, 2, 3, 4, 5, 6, 6 to 8 transfer glucose units, they are transferred to one of this portion and with the help of enzyme transferase. So the next section is how this takes place. This is glycogen primer attached with a few molecules of glucose by 1,4 glycosidic linkage as we discussed. So these are the long chain. Now there is transfer of these 6 units here. So these 6 units are transferred here. 6 to 8 units. And this happens with the help of enzyme transferase. Now there is a difference here. The linkage, the branching point is with the help of this attachment at branching point is with the help of branching enzyme. branching enzyme, and this linkage is 1,6 glycosidic linkage. So basically what? In this glycogen synthesis, there are the three enzymes used. First is glycogen synthase. The second enzyme is transferase, and the third enzyme is branching enzyme. And these three enzymes are required for the synthesis of glycogen. Now, the first enzyme, which is called as glycogen synthase, is responsible for 1,4-glycosidic linkase. And 1,4-glycosidic linkase, so it is responsible for elongation of chain. The second enzyme is transferase, which is required for transfer of a group. Transfer of group. And the third enzyme is branching point enzyme, which is required for the linkage of 1,6-glycosidic linkage. So they all collectively introduce themselves and they ultimately form the complex structure, which is called as, which is a polysaccharide again. This complex structure, which is formed with the help of these three enzymes, is what is a glycogen. And the importance of glycogen in the body, as you know, that in case of fasting, this glycogen can be used as source of energy by converting itself into glucose. So this is about glycolysis. Now the next is regulation. How it is regulated. Regulations are regulation of cycle. This takes place by two things. One is important, that is enzyme, and the other part is hormones. So first I am discussing about the enzyme which is required for this. So enzyme, the most important enzyme is glycogen synthase. This glycogen synthase is responsible for 1,4 linkage and elongation. This synthase occurs in two forms. One is called as active form and the other one is called as inactive form. This active form is non-phosphorylated and inactive form is phosphorylated. Phosphorylated means containing phosphorus and non-phosphorylated means not containing any phosphorus. The hormonal control, as we discussed, is hormone which is responsible for this is insulin. Insulin ultimately causes, including insulin is released from pancreas and its role is to cause formation of non-phosphorylated active form. In case if it increases active form, so it increases glycogenesis. If glycogenesis is increased, ultimately leading to decrease in blood sugar level. So the basic role of uh, insulin is to decrease blood level sugar level by increasing activity of glycogen synthase. So glycogen synthase is called as key enzyme of the cycle. Thanks.